Nivolumab is a human monoclonal antibody that targets the programmed-1 receptor, or PD-1, a key immune checkpoint on T cells. Under normal circumstances, PD-1 interacts with its ligand, PD-L1 and PD-L2, which are often expressed on tumor cells, leading to T cell exhaustion and allowing a tumor to evade immune surveillance. Nivolumab blocks this interaction, reactivating cytotoxic T cells to recognize and destroy cancer cells. It was one of the first PD-1 inhibitors approved by the FDA, initially for advanced melanoma and later for multiple cancers such as lung cancer, renal cell carcinoma, hepatocellular carcinoma, and Hodgkin lymphoma in melanoma. Nivolumab has shown significant improvement in both overall survival and progression-free survival compared to traditional chemotherapy. When comparing nivolumab with ipilimumab, the difference lies in their targets and immune mechanism. Ipilimumab blocks CTLA-4, an earlier checkpoint in T cell activation pathway, while CTLA-4 inhibition broadly enhances immune activation. It also increases immune-related adverse effects. In contrast, PD-1 blockage by nivolumab acts later in immune response, offering a more targeted reactivation with fewer severe toxicities. Combining therapy of nivolumab and ipilimumab is also widely studied. It harnesses the synergistic effect of blocking two checkpoints, resulting in stronger and more durable anti-tumor responses. Though at the cost of higher toxicity, overall, nivolumab represents a milestone in immuno-oncology, shifting melanoma treatment from cytotoxic to immune-mediated control.